traditional cultivation, and identification.
Different media types. So when we talk about media types, there are 4 different ones that we usually talk about. We have enrichment, supportive, sometimes referred as nutrient, selective, and deferential. So when you're learning about the media throughout the course, keep in mind which type these actually are going to fall into. You have enrichment, which contains specific nutrients for growth and for the particular bacterial pathogen. Supportive or nutrient supports growth of most organisms. So an example of this would be like your sheep blood agar. Selective agar allows growth of some organisms while inhibiting the growth of others. McConkey's is an example of this. It allows growth of your gram-negative, but it will inhibit the growth of your gram-positive. And then you have differential, which allows certain metabolic or culture characteristics to distinguish one colony from another. So McConkey's is actually an example of this as well. So it's both differential and selective. So with McConkey's, we can determine if an organism is able to utilize carbohydrates. So we would look to see if it is a fermenter or a non-fermenter. So when we talk about environmental requirements, we talk about aerobic, which means that the organism grows in the presence of oxygen. Facultative, so they're able to grow in the presence or absence of oxygen. And anaerobic, these organisms will be killed if they're exposed to oxygen. So they are basically unable to use the oxygen. So they use a fermentative process. So that's why if you're setting up anaerobic cultures in your facility, you have to have strict protocols just for your anaerobes. And then you have your microaerophilic, which is going to, they require a low level of oxygen. And then you have your capnophilic, which are organisms that grow best with a higher level of carbon dioxide. Most organisms are going to grow best at body temperature, which is 37 degrees Celsius. So we typically maintain our incubators between 35 and 37. When you're doing your clinicals and when you're working, you'll do daily temperatures on the refrigerators, incubators, heat blocks. Campy or Campylobacter jejuni is an example of an organism that actually likes it a little warmer than body temp. So if you're doing a culture of Campy, you're going to have an incubator basically just for that, and it's going to be a little bit warmer at 42 degrees Celsius. And then you have Listeria and Yersinia, who actually grow better in a colder environment. So they are going to be between 20 and 40 degrees Celsius. So room temp is usually about 20 degrees. So anywhere between there, and they grow better. So when we strike our plates, we're going to strike 2 different ways. What we would do is you'll go for isolation. And again, you're going to refer to the figure in your textbook and the video on the slide as well. So typically you're going to think of your plate as divided up into 4 different quadrants. You're going to inoculate typically the upper left with a swab or maybe with a loop if you're thinking like a fluid or spinal fluid. You're going to do the initial streaking, and then you're going to rotate the plate in your hand and finish streaking out the other 3 quadrants. Basically, what your goal is you're trying to get isolated colonies. So that first quadrant you did is probably going to have the most growth because that's going to be the most concentrated, right? And then from there, you're doing the next one from it, and then the next one, and then the next one. So it should theoretically get to be less growth as you go. When we get into urine cultures, they are the only ones that are routinely done for colony counts. Remember, you must use a calibrated loop that delivers a specific amount of specimen. Basically, what you're going to do is you're going to streak for colony count, so a single straight line down the center of the plate. And then either with the calibrated loop or a non-calibrated loop, you're going to come back across that straight line and down the streak, back and forth, so it's perpendicular. Some people call it the Christmas tree look. And just make sure that you're looking at your SOP to see what your facility actually states to do, as they can differ. So when we talk about components for organism identification in a traditional culture method, we're going to look at microscopic morphology and staining characteristics. Again, that most common stain is going to be your gram stain. So, you're looking, are you getting a gram positive or a gram negative? Is it the shape of a coccobacillus? Or maybe we can't tell it's a coccobacillus because it's pleomorphic, meaning it has many different forms. So you can refer to your textbook for colony morphology. You're also going to look to see, are they small, large, spreading, domed? This is once you get it onto the plate. Um, do they have feet? You'll understand when we talk about yeast, when I'm talking about here when I say feet. Do they swarm? Again, what does their outer rim look like? What does their margin look like? What color are they? You're going to ask these types of questions whenever you are looking at your plate. You're going to look at what type of environmental requirements also needed for it to grow. The SOP should indicate the need to incubate those plates. And then again, part of what we'll include with organism ID is if there's a resistance or susceptibility to any antimicrobial agents. There are lots of other tests we can do to try to figure out what organism we are working with, and some of them are enzyme-based tests. And we refer to these generally as a rapid test. So while you do need an organism that's growing, it is something that oftentimes just takes a few minutes to do. So if you know that you've got a gram-positive cocci, growing on your blood agar plate, other than gram staining, one of the easiest ways to differentiate a staph from a strep is the catalase test. This is basically just hydrogen peroxide, and it will emulsify a colony. So basically what you do, sorry, is you'll emulsify a colony in the hydrogen peroxide. And what we're looking for is if it creates any bubbles. So if we get extreme bubbling, that's going to be considered catalase positive, and that's going to mean that we're probably looking at a staph. If you get minimal or no bubbling, that will generally take you to the strep side. So it's a good test for differentiation of those two organisms. An oxidase test can be used to differentiate with our gram-negative bacteria. An indole test is going to be used for the production of tryptophanase, and this is a product of E. coli. So we can kind of determine the different, if it's that or not, and that would be from a gram stain. If we have a gram-negative rod, we can determine it from that. The urease test will assist us in the identification of Proteus species, and a PYR can assist in the identification of strep pyogenes and some Enterococcus versus the other streps that are negative. And we'll discuss these procedures later on in the course, but this is just a quick overview. And then we do have a plethora of commercial systems. So whether this is something like a Vitek or a Microscan walkaway, you will have inoculated the well. It is part of that panel that will include biochemical reactions as well as antimicrobial drugs at different concentrations. So basically, you're going to use the biochemical reactions for identification of the organism, and then you're going to use the antimicrobials to determine the sensitivity or resistance of that specific organism for each of the drugs. So if your biochemical reactions can be based on the colorimetric or fluorescence or turbidity. So basically with turbidity, it may be clear, but we're looking for cloudiness or haziness that indicates growth in that well. And antimicrobial susceptibility testing typically is based on growth or no growth. In the lab world, we always have a control well, and we're going to compare that as well. So what we report with the antimicrobial susceptibility testing is referred to as a minimal inhibitory concentration, or an MIC. The minimum concentration of that drug that will inhibit, not necessarily kill, but inhibit the replication of that organism. And then from there, we'll give that report to the physician, who will then be able to know what to prescribe for their patient, which will help get rid of their disease or infection.